Life Sciences, Week 4, Video Lecture 9, Organic Compounds, DNA, RNA, and ATP. Okay, so this is our last video on organic compounds. And so what we're looking at in this one are the DNA and RNA molecules and the ATP molecule. So let's take a look. So the first thing we have here is a little history of the structure of the discovery of the structure of DNA. So on the right here, we have Rosalind Franklin. Rosalind Franklin is the chemist who discovered the structure of DNA. But she was one of these pioneers in the world of science, and being by pioneer I mean one of the few women in the world of science at the time. And so she worked with the scientist to her left, who is Maurice Wilkins. Maurice Wilkins thought that she was his assistant. So he thought that, and she was not hired under those circumstances. She, believed that he and Wilkins were colleagues on equal level. So anyway, so it was his belief that any discoveries she made, since she was working under him, were his discoveries, essentially. So anyway, so Rosalind Franklin, along with a student of hers, did work with they froze DNA strands and then looked at the X-ray diffraction of those crystallized DNA molecules, and so that makes a very distinct X-ray signature that they then could use to extrapolate from that the structure of the DNA molecule, so that like the twists and turns and what degree those were at and that kind of thing. So going into that work, she already knew what DNA was made of, because that's easy enough to put it in a blender and mix it up and see what's in there. And so she knew that's she didn't exactly put it in a blender, but you know what I mean. So she knew that there was phosphate. She knew that there was ribose. She knew that there were bases, adenine and guanine and cytosine, etc. So what she was after was figuring out the structure, so how these things were all connected to each other to create this DNA molecule, and that's what she used her X-ray diffraction to figure out. So she presented her findings at a lecture, and one of the people attending that lecture was James Watson, who is the next person to the left there. James Watson worked with his colleague Francis Crick, who is the guy on the far left, and they for some time had been working on figuring out the structure of the DNA molecule. So after attending her lecture, he left and would continue to work with Francis Crick, and basically these two researchers, Watson and Crick, they were not so much in the lab kind of researchers. They were compiling information from other people. So they were looking at the research other people had done and taking that information and putting it together and seeing what seemed to fit and what didn't seem to fit. And in that process came up with their theory on the structure of the DNA molecule. Once Maurice Wilkins showed them Rosalind Franklin's the image that's on the top of your notes there, the X-ray diffraction of DNA, that essentially confirmed their conclusions about what the structure of DNA was. And they published a paper the exact same year, actually, that Maurice Wilkins published a paper with Rosalind's work. And so both papers were basically saying the same thing, which is what the structure of DNA is. So Watson and Crick are the ones who are officially given credit for discovering the structure of DNA, but the Nobel Prize for that, about 9 years later, went to Watson and Crick and Wilkins, those three. And the Nobel Prize, people say, oh, well, we don't award Nobel Prizes after someone has passed away. So Rosalind Franklin actually had passed away by that point and did not receive a Nobel Prize or recognition for her work. So partly that goes to show that it takes a lot of people to figure out something very complex like the structure of DNA. Figuring out that structure went all the way back to starting with in the 1870s. German anatomist Walter Fleming is the one who discovered DNA at all. So he discovered that that there were these thread-like, fiber-like things inside the nucleus of cells. And so that kind of started the research in what are these thread-like fibers. And and little by little we built on all that information until we found out what atoms were in there and how they were likely arranged and et cetera, until we got around to the 1950s, which was when the structure of DNA was discovered. So that's a little history on DNA and a little sort of snapshot of not everything is always fair in the world of science. This is Franklin's X-ray diffraction image of DNA, and I don't know how to read X-ray diffraction, so I can't really say that much about it, but she was able to figure from this the shape of the DNA molecule, and also where the phosphates and sugar molecules were in relationship to the base pairs. All right, so let's take a look at this molecule, deoxyribonucleic acid, or DNA. So this is what DNA stands for. The D is the D of deoxyribonucleic acid, the N is the nucleic, and the A is the acid. So, early on in the discovery of DNA, we were able to tell that DNA molecules behaved as acids. In other words, if we put them into solution, they donated hydrogen ions to the solution. And they were found in the nucleus of atoms, and so that's where the term nucleic acid comes from. Nucleic in the nucleus, and acid, it donates hydrogen ions to the solution. So DNA specifically, our first nucleic acid that we're looking at here. DNA, or deoxyribonucleic acid. So the functions of DNA in the body are that it contains our genetic material, our inherited genetic material. So every single cell in our body, except for cells that don't have a nucleus, all the rest of our cells have a total complete copy of all 23 pairs of chromosomes. And let me just mention, so in the notes there's a diagram, I don't think I have it here, no. In the notes there's a diagram of the 23 pairs of chromosomes. The chromosomal form of DNA is only a temporary form that it takes during cell division. The rest of the time, DNA is like a bowl of spaghetti in the cell. It's all strung out, and which is what it needs to be for enzymes to be able to access the genes to get the information from them. Any given cell, so say we're talking about a liver cell. A liver cell contains all of the DNA, which contains all of the information on how to make every single protein in the whole entire body, and regulatory information about when to make those proteins and what quantity to make those proteins in, and all of that. But a liver cell specifically will only utilize the genes within all of the DNA that have to do with being a liver cell, building and running and operating a liver cell. Whereas a bone cell will only access the information that it needs about how to make the structures of a bone cell. An eyeball cell will only use that part of the DNA, etc. So even though all of the cells have a full copy, they all will only use a tiny part of it based on what they need to make. So DNA, each DNA molecule is divided into sections called genes. One gene is a stretch of DNA that contains the code for how to assemble one single protein. So, and we actually have many copies, or a handful of copies, of many of the same genes. So say we're talking about the protein collagen. That's a great example because there's so much collagen in the body. Collagen is a protein, and it's a protein that shows up in collagen fibers show up in all of our connective tissue in the body. So we're going to actually have multiple copies of that gene on how to make collagen, and when we have cells that are actively making collagen, we'll probably access all of those copies of that same gene so that we can make larger quantities of it in a shorter period of time. But the idea, though, is that each DNA molecule contains specific genes for specific proteins, or specific regulatory genes about when and how much, etc., to make certain proteins. And with that information, we can build our whole body. So different DNA molecules have different genes, contain different genes on them. And let's see. So what we're looking at here on this slide is the double helix structure that Rosalind Franklin proposed was the structure of DNA. So if we imagine this as a ladder that we've taken and we've twisted it, that kind of gives us a good visual of what we're looking at. It's this twisted model. It's considered or the model is called the double helix model. So it's a double-stranded molecule, but there are twists to it. And so if we consider it like a ladder that has been twisted up, there are two parts to it. There are the rungs of the ladder, the parts that your feet step on as you're climbing a ladder, and those are across the middle. And then there are the sides of the ladder, the part that your hands hold on to. Well, although I guess your hands could be on the rungs, but the sides of the ladder. So on this slide, the part that shows green. Okay, so we're going to look at each of those two structural components of the DNA separately. We'll start with the rungs of the ladder. So the rungs of the ladder of the DNA molecule are made up of 4 different nitrogenous bases. So nitrogenous bases, what are those? What does that mean? Nitrogenous implies that they have nitrogen in them. And if we look at these, can I zoom in? Oops, sorry, strange things are happening here. Come back. Okay, wait, let's go back. All right, here. So if we look at these molecules, so the blue hexagon, the black hexagon with a gray pentagon attached to it, the green hexagon with a gray pentagon attached to it, and the red hexagon, those are the four different bases. So or nitrogenous bases. So they all contain nitrogen, that's the nitrogenous part of them, and they're all bases, meaning, so if we remember what happens with a base when we put it in water, it either donates hydroxide ions to the water or it pulls hydrogen ions out of the solution. So that's the behavior that these guys do, and so that's why we call these nitrogenous bases. So we have 4 different nitrogenous bases that make the rungs, but each rung is made of 2 of them put together. And then hydrogen bonds form between them. So they have to pair very specifically. So on top here we see cytosine pairing with guanine. So cytosine and guanine pair with each other, and there are 3 hydrogen bonds that go across and form that connection between the cytosine and the guanine. And then the other pair are thymine and adenine, and there are 2 hydrogen bonds that form between those. So it's the hydrogen bonds that hold the 2 sides of the 2 strands of DNA together. So those are the rungs of the ladder. And so every rung of the ladder is made of a pair of these bases. So it's either adenine and thymine paired together or guanine and cytosine paired together. Okay, so that's the rung part. So the other part of the DNA molecule are those sides of the ladder. And so those sides of the ladder are made out of sugar, and let me see here. So I think that's our view of the whole thing. We can see the sugars here though too. So if we look at, for instance, guanine is that black hexagon. It has a gray pentagon attached to it, and then attached to that is just a clear pentagon. That pentagon is a sugar molecule. It looks kind of like fructose. When we were looking at monosaccharides, we saw that fructose was one of the monosaccharides that shows up in a pentagon shape. This one is called ribose, but it is a monosaccharide, and it's the monosaccharide. It's a structural monosaccharide that we use to build ATP. To build DNA. So it's actually in DNA, it's not called ribose, it's called deoxyribose. Sorry, not to confuse things. So anyway, that sugar molecule is called deoxyribose, and that's where the first part of deoxyribonucleic acid comes from, is from that sugar part of it, which is part of the side of the molecule, deoxyribose. Okay, so between the neighboring sugar molecules, those deoxyribose molecules, there are phosphate groups. So you can see the phosphate group that is a P with 4 oxygens attached to it. That's a phosphate group that connects each of the neighboring monosaccharide, deoxyribose molecules to each other. So if here we look at a little bigger stretch of DNA, in the middle of this diagram, we can see the base pairs pairing across. So we see adenine and thymine at the top with their two hydrogen bonds between them. The next one we see thymine and adenine, so they're just in different locations, but still two hydrogen bonds connecting them. And then we see cytosine and guanine with the 3 hydrogen bonds between them, and then flipped around we see guanine and cytosine with 3 hydrogen bonds between them. So that's the joining of the two sides, and those are the four different bases that we see, the nitrogenous bases that we see in DNA. And then we see the blue deoxyribose molecule and the phosphate groups along the sides. And then if we kind of zoom out from there, then we have the image that we're looking at on the left, where those blue sort of spirally strands are this, you can see it just has SP, 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 so it's sugar, phosphate, sugar, phosphate, sugar, phosphate. That's what the whole side of it is made of. And then across the center we see the T's bonding with the A's and the C's bonding with the G's to make the rungs of the ladder. So that's our structure of the DNA molecule. So we're not going to get into deep groovy detail right now about the information that's held on a DNA molecule beyond what I said earlier, which is that each gene on the DNA molecule has the information on the primary structure for a single protein that the body makes. But let's go on and take a look at the structure of ribonucleic acid, which is RNA. So there are a few differences between DNA and RNA. So here we can see both a DNA molecule and an RNA molecule. So let's take a look at these differences. So they're very similar. Their names sound very similar, so that means structurally they're very similar, but let's take a look at what's different. So RNA is a single-stranded molecule rather than a double-stranded molecule like DNA. So DNA is double-stranded. However, only one of the strands is the strand with the coded information on it. The other strand is basically there to stabilize the molecule, because it makes it a sturdier molecule for it to be double-stranded. It protects that coded information in a sense. And so it is double-stranded so that it is sturdier and more protected, but only one of the strands of the DNA contains that coded information. RNA molecule is single-stranded. So it's single-stranded partly because it's a temporary molecule. So it's okay if it's a little, if it's not as sturdy as DNA, but also it's more of a, like, we can think of DNA as a molecule that stores that genetic information, whereas RNA is part of the working machinery of building the protein. So the information on the RNA molecule needs to be more readily accessible. So it needs to be a single-stranded molecule so that it can be read and proteins can be assembled from it. So single-stranded molecule RNA is, and then DNA is a double-stranded molecule. RNA is formed inside the nucleus, right where DNA is, but then it leaves the nucleus after it's formed. So DNA is always going to be inside the nucleus. Unless the cell is dividing and the nucleus goes away. So during cell division, the nucleus will dissolve for a while cell division is happening. But as long as there's a nucleus, DNA will be inside of it. RNA is built off of DNA, and that happens inside the nucleus. But once an RNA molecule is formed, it will leave the nucleus, and it will do the rest of its work out in the cytosol outside the nucleus. So so even though these are both called nucleic acids, and that term comes from the fact that they're found in the nucleus of cells, RNA is not always in the nucleus of cells. It's sometimes in the cytosol, or a lot of times it's in the cytosol. It's only formed in the nucleus. RNA is a much smaller molecule. So DNA, if we took one strand of DNA, even the very longest strand of DNA we have, it may contain hundreds and hundreds of genes. So the coded information on how to make hundreds and hundreds of different proteins to make our body. RNA contains only the information for one of those proteins. So we're just going to make an RNA molecule we'll use the DNA as a template, essentially, and make an RNA molecule using the DNA as a template, but we're only going to read one gene's worth of material. So we'll make a copy of that information on how to make that one gene, and then that's what's contained in the RNA molecule, and then the RNA will leave the nucleus, and that's what's going to happen is we're going to make that protein that the RNA has that information for. So it only contains the information for one protein, whereas DNA has hundreds and hundreds of genes. RNA is a temporary molecule, so after we make the protein that RNA has the information on how to make, then we're going to break it down and get rid of it. So it's only going to be around temporarily while that protein is being made. And then when we take it apart, we'll disassemble it and we'll recycle it. We'll most likely send the parts back inside the nucleus where they'll get recycled into another RNA molecule as that cell needs to make some different protein. Okay, another difference between DNA and RNA, the sugar that forms them. So structurally, they're different in a couple of ways. So remember we talked about the sugar that's part of the backbone in DNA is called deoxyribose. In RNA, the sugar that is between the phosphates that forms the backbone of the molecule is called ribose. So, so there's a slight difference in that sugar molecule that that helps to hold the whole thing together. And one other structural difference is we had talked about DNA has 4 bases: thymine, cytosine, adenine, and guanine, and that's true for RNA too. It also has 4 bases, but there is no thymine in RNA, and instead uracil is what pairs up with adenine. So so that just looks a little bit different. So like on this diagram that we're looking at, thymine is red, and wherever there's thymine across from it is a yellow adenine. So on the DNA molecule, or sorry, on the RNA molecule, if we're going to make an RNA molecule using that DNA template, wherever there's an adenine on the DNA, we're going to put a uracil on the RNA. So we still have to pair them up like that. So wherever there's a cytosine on the DNA, we're going to put a guanine on the RNA. And wherever there's a thymine on the DNA, we're going to put an adenine on the RNA. So everything has to pair up, but there is no thymine in RNA. It's uracil instead. So if it's time to pair up with an adenine, it's uracil that's going to be put in that place to pair up with it. Okay, so those are the differences between DNA and RNA. And okay, and I think that's it for the nucleic acids. So let's go on and look at our last molecule here, which is adenosine triphosphate. So adenosine triphosphate I've referred to, especially when we were talking about carbohydrates. We were talking about carbohydrates being broken down to be used as fuel to make ATP, and that's what this molecule is, ATP. So ATP, adenosine triphosphate, is our energy exchange molecule in the body, or our energy currency, as it's often referred to as. So let's see here. Let's look at our functions of ATP. So we can make ATP from a number of different fuel sources. So carbohydrates, proteins, lipids, any of those macromolecules, we can break them down, and we can take their monomers and break those down and break the bonds and release the energy in those bonds and essentially harvest that energy and put it into an ATP molecule. So we're going to get different amounts of energy out of those different molecules because they don't all have an equal amount of energy stored in their bonds. But carbohydrates, lipids, and proteins all can be used as fuel to make ATP. But that's the thing, the body needs whatever it is that we're using for fuel. It has to be converted into that ATP molecule because that's the only one. That's like, it's kind of like, oh, like if carbohydrates were pounds and lipids were francs and proteins were yen, right? So 3 different types of currency from around the world. But we want to spend it all in the US, so before we can spend it, we have to switch it to dollars. So that's essentially what we're doing. We're taking different fuel sources and switching it all into the same molecule, which is the only one that we can actually use to do the energy transfer in the cells. So that's the ATP molecule. So functionally what ATP does is it transfers stored energy. So remember, the energy stored in chemical bonds, we can call that chemical energy. So that's how a molecule can store energy for later, is by putting it in its bonds. So that's chemical energy. So what ATP does is it transfers chemical, yeah, I guess transfers is the word. It changes chemical energy into kinetic energy. And so if we remember, kinetic energy is the energy of movement. That's essentially what ATP is doing, is changing stored energy into moving energy, but on a molecular level. So let's look at the structure of this molecule here. So on the top right, we have one adenine. And so adenine, at this point, should sound familiar and look familiar. It has a bunch of nitrogens in it, and it has a hexagon and a pentagon put together. It looks a heck of a lot like the adenine from DNA and RNA, and in fact it is. It is the very same adenine. So we have one adenine molecule in ATP, and that is bonded to one ribose. And ribose also should sound familiar. It's a pentagon-shaped monosaccharide, and it is the exact same pentagon-shaped monosaccharide that we find in RNA, making that backbone of RNA with the sugar-phosphate sequence is a ribose molecule that we find in RNA. So those two things are two things that we've seen already, because that's part of the structure of RNA. Now attached to that ribose, we have three phosphate groups. So the first phosphate, the one that attaches to the ribose, that's the first phosphate. The middle one is the second phosphate, and then the third group is the third phosphate. So it's that third phosphate that is actually kind of hard to attach when we're making an ATP molecule. And so it takes a fair amount of energy to make that, to create that bond. That energy that it takes to create that bond, that is the energy that we're harvesting out of the molecules that we're breaking down to make this ATP molecule. So whether it's a glucose molecule that we're breaking down, or a fatty acid that we're breaking down, or whatever it is, the energy that we're getting out of that, we're going to essentially harvest that and put that into forming that bond. And we call that bond a high energy bond. So that's the bond between the third phosphate and the rest of the molecule. It's a high energy bond. So what happens when ATP does its thing? When it transfers energy to another molecule. So here, we actually, on this diagram, have flipped the ATP around, but we can see the blue adenine, so we're looking on the left here. We can see the blue adenine connected to the pinkish ribose, and then we see 1, 2, 3 phosphate molecules, but we see sort of a sunshiny kind of shape at the bond between the second and the third phosphate. So that's indicating that that's our high energy bond there. That's the one that's really storing the energy. And then when ATP goes through a reaction and transfers the energy from its high energy bond to another molecule, what happens is that third phosphate breaks off. And so on this, you can see is labeled as inorganic phosphate. So that's a good example of organic and inorganic chemistry interacting, because ATP is an organic molecule, but it could not do what it does without the inorganic parts of it. So phosphate is a very important part of ATP. So what happens is we break that high energy bond, that third phosphate comes off, it transfers actually to, in many cases anyway, it actually attaches to and transfers to the molecule that the energy is getting transferred to, and in the breaking of that bond, energy is released. But the other thing that happens when that energy is released and that bond is broken is that energy gets transferred to another molecule, and in response, the other molecule, the one that's receiving the energy and receiving the phosphate, it changes shape. And you know, molecularly changing shape doesn't seem like a very big thing. You know, it's not. On a macro level, one molecule changing shape is not a very big thing. But on a macro level, we can have bazillions of molecules changing shapes, and that creates the big movements of our body, and it also creates the small movements of our body, or the things that are happening on a cellular level that we can't feel or detect and don't have a conscious sense of these things happening. So let's take a look at ATP in action. Let me just glance at the notes to make sure I didn't leave anything out. Let's see. Okay, so yeah, let's just take a look then at ATP in action. So this diagram is showing one little tiny twitch of a muscle contraction. And we're actually going to start on the bottom of the diagram where we see ATP, that sort of sunshiny shape ATP coming into the equation. So ATP is coming in and is attaching to that sort of balloon-looking molecule there, which is a myosin molecule. So myosin and actin are two proteins inside muscle cells that slide past each other for a muscle contraction to happen. So what we see is ATP coming along and attaching to this molecule called myosin. Myosin is a protein. So it attaches, and when it attaches, it actually, in this case, it instantly transfers its energy to myosin. And so if we follow the arrow up to the left, what we see attached to that myosin molecule now is ADP and P as two separate things. So when ATP transfers this energy to another molecule, it turns into ADP, adenosine diphosphate, plus the separate phosphate that came off. And that's what we see has happened. And if you'll notice, between that bottom diagram and the one that's like in the 9 o'clock position up to the left of it, you can see that the myosin molecule is shaped differently in those two diagrams. So as the ATP transfers its energy to myosin, that changes the shape of myosin. But ADP and that third phosphate are still attached to the myosin molecule at this point. So if we then continue clockwise up, and now we're at the 12 o'clock position, at this point now in the process, now that the ATP has transferred its energy to the myosin molecule now has part of its shape change that it went through was that binding sites were revealed on it that will allow the myosin to bind to that molecule above it. So the molecule above it is made out of a protein called actin and a couple of other proteins, but that's basically a fiber formed out of proteins. But so once the ATP transfers its energy, the myosin now is attracted to, it has changed shape to reveal a binding site, and so now it attaches to the actin molecule. So now we're ready for a muscle contraction to happen. And so or a twitch, which is a tiny little microscopic aspect of a muscle contraction. So in order for that, so the thing that happens next, once the actin and myosin bind to each other, ATP goes, well, which is actually ADP plus P, so they're separate here, but they go, oh, myosin, you're attached to actin. I actually don't even like you anymore. Goodbye. And so, but basically what's happening is once the myosin attaches to the actin, the binding sites for that ADP and the P go away on the myosin. So that essentially kicks off the ADP and the phosphate. So when they go away, you can see that another positional change happens in the myosin molecule. So now it sort of like bends its balloon head over, and since it's attached to the actin fiber, it pulls that actin fiber along. So what have we seen happen here? We've seen ATP attached to a molecule. The attaching of ATP transferred the energy to that molecule, which changed the shape just slightly of that molecule and revealed a binding site on that molecule. It binds, that gets rid of the ADP and the phosphate, and then another change in shape happens to that molecule, and that's the twitch, or a molecular aspect of a contraction. So if we have this happening enough times and in enough molecules all at the same time and in the same location, this is going to switch from a twitch, or one tiny little change in the shape of one protein, into a whole entire muscle contraction, so a movement of the body. So this is this tiny molecular thing that when we get enough copies of it happening all at the same time, we get big macro movements that are identifiable in the body. So that is ATP. Oh, I don't have another slide on here. So let me just summarize your homework. After this is nothing. You don't have homework that is specific to this lecture, focusing on the nucleic acids and the ATP, but we did in this lecture look at the structure and the function of DNA and RNA, and we looked at the structure and the function of ATP in the body. So rather than doing specific homework for this, your next homework is to complete exam 2, which is up for you on Populi and should be ready for you to take as soon as you are ready for it. So that's due by class time next week, and text me if you have any questions. And let's see, anything else? And then next week, then we start to get into plants and microscopic life forms. Okay, so we're on to I think that's next week. Anyway, we're on to bigger things. We're kind of moving out of molecules and starting to get into cells and larger living systems. All right, that's it for this lecture. And let's see, anything else? Good luck on the exam. I'm sure you'll do fine. I hope all of this is clear, and I'll see you in class next week.